biotechnology, the DNA technology that we have today, and I'm actually going to go through all of the DNA tools that we have. So there are a million different things that we can use genetic engineering for. And one of the things I spend a lot of time teaching is we now actually have the capacity to change human genetics, which you're gonna be working on an activity for that this week, actually. So we are looking at the human genome, which is 3.2 billion bases. Um, we figured that out about 20 years ago, right? Okay, so genetic engineering is a manipulation of DNA. So we're going to go through this set of tools that we have, starting back from like the beginning. 20 years ago, this was a big deal. Now the things that I'm talking about, not so much of a big deal, and I'll continue to just kind of go through and tell you what is important to know. So I am going to talk about bacteria. We haven't talked about prokaryotic cells yet. We will be spending a lot of time talking about that soon. You work with bacteria, you work with bacteriophages. Bacteria are one-celled, single-cellular. They're prokaryotic, which means that they do not have a nucleus to hold their DNA, so their DNA is just kind of out in the cell. They also don't have membrane-bound organelles like the Golgi apparatus and the smoothie are. They reproduce through binary fission and they grow very rapidly, which makes them a very good tool. They are the dominant life form on Earth and they are incredibly diverse. So they have a cool genome, which is much different from ours. Our DNA is organized into chromosomes wrapped around histones, right? They have this single circular chromosome that's haploid, which means half of the DNA. It's naked, no histones, about 4 million base pairs, 4,300 genes, okay? So we have talked about transformation before. When Griffith decided to kill a bunch of mice and realized that there's some sort of transforming factor, he wasn't able to figure it out because he was killed. But one of the things that we like to do in the lab as high school teachers, and I don't know if you guys have done it in college, is something called transformation. So bacteria are opportunists. They'll pick up naked DNA wherever they go, right? They have these surface proteins that are specialized for uptaking this DNA. And they import bits of chromosomes from other bacteria. They incorporate it into their DNA, and then they express these new genes. It's called transformation. Okay, and it's a form of what we call recombination. Recombination meaning different arrangement of genes. This is really easy to simulate in the lab. A lot of times, I'm going to go back to this. You can make what's called competent cells out of E. coli. We use E. coli a lot. We put it in calcium chloride. Calcium chloride makes holes in the cell membrane, so it will uptake more DNA. A lot of the things we like to do is make bacteria glow in the dark using a jellyfish gene, so we incorporate a jellyfish gene into bacteria. I have one more recently I like to do that bacteria can be colors of the rainbow, right? So what are plasmids? Plasmids are really important in the use of biotechnology. Bacteria have these other DNA, so this is the regular DNA for bacteria, but they have these small circular pieces of DNA called plasmids that they exchange with the environment and they exchange with each other. They carry genes, but this is where you find genes for antibiotic resistance. So these things are so easily exchanged between bacteria that it causes a very rapid evolution of bacteria. They're constantly changing, constantly changing. Can also be imported from the environment. So if we have one bacteria that picks up a gene for antibiotic resistance, you better be sure that all the bacteria around it are going to also get that resistance because of these small plasmids. Why is that important? We like to use plasmids a lot because they can get into bacteria easily and because we can change them. We can manipulate them really easily. We can recombine them. In the lab, we can change the DNA and then we can put it into a cell and we can make it inject its DNA to something else. We call it a vector. So I do want to just point out that there are a lot of vectors. Plasmids are an older vector. We now can manipulate viruses to be vectors. One of the most popular viruses that we use is called the adeno-associated virus. That is a great vector. We're using retroviruses. We're using the herpes virus. We're using the adenovirus, which is a cold. So when we talk about vectors, we talk about taking out, especially when we're using viruses, we take out the disease-causing gene and putting a gene in that we want to change, right? So gene therapy is something that's really important that you're going to be learning a little bit about in a minute. But we use these things called vectors. A lot of times they're viral vectors. Plasmid vectors are limited. Okay. So again, like I said, plasmids can insert new genes into bacteria. So genes we want: insulin, HGH, and lactase. Okay. All right. We use ligase to glue them together. Ligase should be a refresher for you, and this is recombinant plasmid. Okay. So really quickly, I want to talk about something called PCR. You may or may not have done this in the lab. So PCR, polymerase chain reaction, is how we can make copies of eukaryotic DNA when we need more. We essentially are forcing DNA to replicate in the lab. It's used to amplify DNA at crime scenes or to amplify a gene of interest for study. This essentially is what happens. I'm pretty sure you guys have done this. You put samples of DNA into what we call our PCR machine, and all of these things happen, right? We denature the protein. We add DNA primers and DNA polymerase, usually Taq polymerase. So at 95 degrees, the DNA denatures, means it unwinds. So we're forcing it to unwind, do helicase's job. Step 2, at 55 degrees, we have we put in DNA polymerase and we put in all these nucleotides. So now we're rebuilding the DNA. So step 3, they're synthesizing at 72 degrees. We found the correct temperature to do this. And then step 4 is just finishing this DNA. And we repeat this cycle 20 to 40 times to get more DNA. PCR just simply means polymerase chain reaction. We add polymerase, we add primers, we change the temperature.
Good morning. Please excuse the interruption. As of right now, the elevator is not working. We will turn
Sorry
that
about
on
this.
when it is working, but right now it is not working.
Anyway, so we do this 20 to 40 times to get more DNA to study genes of interest, right? So the PCR COVID test, all that was doing was amplifying DNA, trying to find if we had DNA of COVID-19, right? They put in polymerase and then test. Is it that? However, they repeated it an obscene amount of times, so I don't know what the point of that was, but they did. Anyway, so DNA needs to be cut. We cut DNA with what we call restriction enzymes. They're endonucleases. I know that I put something on your quiz about this before I actually taught you about it, so I apologize. This was discovered in the 60s. Really, what this is a bacteria has these bacteriophages, as you know, and they kill it. So they have their own immune system called restriction enzymes that will go in and cut these enzymes up, or cut the DNA of the viruses. So viruses will insert their DNA, and then they'll send the restriction enzymes up to cut their DNA up so it's useless, right? They restrict the action of attacking an organism. And you're going to learn about CRISPR. CRISPR-Cas9 is a form of a restriction enzyme, right? So the bacteria protect their own DNA, right? Um, so they cut at specific sequences. We call them restriction sites. There are symmetrical palindromes. So if you take a look here, it's GAATTCC, GAATTCC. So this particular enzyme that we're looking at will cut in between the G and the A at this location. There's many different enzymes that we use. We have EcoR1, HIND3, BAM, HI, and SMAL1. Again, you're looking at what we call the restriction site. So as a result of this cut, we have something that's called sticky ends, and we can do a few things with these sticky ends. Okay. And I'm going to talk about a few of them here. So the first thing that we can do after we cut our DNA with restriction enzymes is load it into a gel and apply electricity. And what will happen here is it will separate DNA pieces. DNA will travel down this gel, which is more of like a very porous type of jello, right? You can see all these bandings. I'm pretty sure you guys have done this before, but the reason this happens is because we apply electricity here. DNA has a negative charge because of its phosphate groups. So if we put the positive charge down here and apply the electricity, it runs the DNA down this gel, separating it out into different fragment sizes, right? So if we take a look here, Here's our power source. We have the negative electrode. We load our DNA into here. Here's a positive electrode. This is what we call a ladder. We use a ladder to compare, to be able to measure the size of DNA. We know the size of all of these pieces. And then what will happen is this banding will appear based on how the sizes or the fragments of DNA here. And so we have the longer DNA stays up here, the shorter DNA here. Restriction fragment length polymorphisms are a fancy way to say everybody has their own fragments, right? Everybody's fragments are different. If you put my DNA in with some restriction enzymes and then we just ran it through an electrophoresis, mine would look different from yours. So everyone has their own RFLP. It's very unique. It can help identify paternity, crime scenes, diagnose disease. It's very useful. So if we just take a look here, DNA fingerprinting, we're looking at a crime scene. Here's the DNA. So what will happen a lot of times is they'll find DNA, they'll run it through PCR to get amplify it to get many millions of copies of it, and then they'll run they'll run it through the electrophoresis to see the RFLP. So you can see suspect 1 is not the person who did the crime, although they have 2, 3, 4 fragments that are the same. But then if we take a look at suspect 2, it is the same, right? So everybody has a unique RFLPs, and this is what gets people caught in court, okay? The other thing that we can do with sticky ends, right, because we like to do cool stuff with DNA, we can cut other DNA with the same enzymes and leave the sticky ends on both, and then you can glue them together at their sticky ends. So here's the gene you want. Here's the chromosome you want to add the gene to. Here's how we can combine it, okay? So we're taking a look here. DNA ligase, if you throw DNA ligase in with this, it will glue them together. And then we have what's called recombinant DNA, okay? Gene produces a protein in different organisms or a different individual. So we have the human insulin gene in bacteria. Why do we do that? Because we need insulin. So we want to have bacteria produce that insulin for us. This is how we do it. Okay. That's how we get human insulin. The code is universal for all organisms, all living organisms. That's why it makes it successful. So we're talking about transformation. We can insert in recombinant plasmid. I had talked about this earlier. We like to grow. I talked about the glow-in-the-dark, or the ones that we make colorful in the lab just for fun in high school. You can grow recombinant bacteria in these agar cultures, and then the bacteria can make a lot of copies of the plasmid. That's called cloning the plasmid. There's a production of many copies of this inserted gene, and then a production of a new protein. So we call it a transformed phenotype. So in the cases that we do this in the lab, in high school, it's a glow in the dark. All these bacteria glow in the dark, and they're antibiotic resistant. So we've introduced antibiotic resistance and a glow. Okay. All right. So it's this growing bacteria to make more. We take a gene from another organism. We make a recombinant plasmid. We transform the bacteria. We grow the bacteria, and then we harvest and purify that protein. In this case, we're talking about human insulin. So what other ways are we using genetic engineering? We genetically modify organisms. It enables plants to produce new proteins. So genetic modified organisms, like them or love them, our food system is full of them, right? So one of the things we're using it for is we protect crops from insecticides. So we have BT corn. It's also soybeans. I'm going to say about 90% of the soybeans that are made in America are BT. So the corn produces a bacterial toxin that kills the caterpillar that kills corn. And then, we have Roundup Ready soybeans. I apologize. Where you can spray the plants with this Roundup. Glyphosate, which is a hot debate right now. Glyphosate causes non-Hodgkin's lymphoma, and people have actually sued Monsanto, which is now Bayer, and won millions and millions, but we're still spraying it all over our food, something you should probably be aware of. We can also extend the growing season of different organisms, including the tomato that we're working on. So fish berries, strawberries with anti-freezing gene from flounder. We also are using that for tomatoes. We can improve the quality of food. So golden rice, I've been hearing about golden rice for about 20 years now. And I do have to say, it has not come to fruition. It has the potential to produce vitamin A and improves nutritional values for countries that need it, but it has yet to be successful. Um, I tend to be, it doesn't matter what I think, but we are using genetic engineering to modify our food system as our population grows to meet the needs of our population. Okay? So this is the thing that I was telling you about what we can do to bacteria. We use GFB protein in bacteria, but we're also using it in mice and pigs. We're making them glow in the dark. Yes, we are really doing that. We've been doing it for a while. It's really odd, right? But we do, but a lot of it has to do with research and producing medication. All right. So we cut, we paste, we copy, we find. I presented you with some older technology. I'm going to ask you to go ahead and work on the assignment that I've posted for you. It is important that you learn about the new technologies, gene therapy, CRISPR, RNA interference. We're going to discuss these at great length in class.